My name is Chider Chen. I'm from Oral Surgery Department, School of Dental Medicine at the University of Pennsylvania. My lab is mainly working on the mesenchymal stem cells. We find this cell have their unique ability to do the tissue regeneration and also the disease treatment. We'll talk about this later. But the thing is, when we try to translate our finding from cells study to the clinical application, we found it's quite difficult to expand the enough cell number to translate our finding to the real patient study. So that's why we try to find some replacement cell source. Like in this case, we use the TSC as a early stage stem cell, and we try to understand if this cell can help us to expand the cell number and to do the tissue regeneration or the disease treatment in translational purpose. Okay. So what is the mesenchymal stem cell? So the mesenchymal stem cell is first isolated by Dr. Brandstein in 1974. So simply, he just fresh out the bone marrow cells and the culture in the dish. And after overnight, they wash out the unattached cells, and the attached cell will form the stem cell colonies. We call colony forming unit, fibroblast, because this cell, the morphology of this cell are kind of a spindle shape, very similar to the fibroblast. So we call this is the colony forming unit, fibroblast. And they found this cell have very unique ability with their multipotent differentiation ability. They can differentiate to many different kind of mesenchymal lineage cells like bone cell, fat cell, or chondrocytes. And also they have like their self-renewability. Okay. So this, we call this adult stem cell. And very recently, around 10 years from now, we like 10 years around these decades, we found MSC not only a good cell source for tissue regeneration, for this, they also can interplay with immune system. So we can use this cell to cure immune-related diseases. In this case, MSC can interplay with many kinds of different immune cells like T cell, B cell, macrophage, neutrophil cells. But we are more focused on the T cell, so we try to use this cell to cure pro-inflammatory disease like peritonitis, like osteoporosis, like autoimmune diseases. So this is kind of a MSC background. These cells are very unique. They can have tissue regeneration ability. They can also use for some disease treatment. But again, as I mentioned, when we try to do expansion of these cells, we find that the cells are easy to become senescent, or we call cell aging, because during the in vitro continued culture, we found there are a lot of change of this adult stem cell isolated from the mesenchymal tissues. Okay, so this involves a lot of regulations during the in vitro cell culture. I don't want to go too much detail, but it seems like their genetics status and also the like mitochondrial function all decrease. So this causes the stem cell function decrease and cause the aging of the cells. So it's make us very difficult to translate our finding to translational application. Okay. And also, if we're looking for the in vivo stem cell status, we found the stem cell, no matter the number or function, they are very highly activate during the developmental stage and maintain in a certain stage in the adulthood. Then they also go quickly going down in the aging status. So it's also required to have cell source to replace the senescent stem cell in vivo for tissue regeneration and for some disease treatment. Okay, so that's why we start this project with the company to use the TSC as a replacement cell source for mesenchymal tissue regeneration. And then I think in this meeting, most the people in this meeting are kind of experts in this field. So I don't want to go too much detail in the background information of TSC. But right now, in the literature, most of people believe the TSC can mainly differentiate to the placental developmental tissues. But largely it's unknown if they can go to other lineage like a ESL, they can go to 3 lineage like ectoderm, endoderm, and mesoderm. How the TSC can go to different lineage cells are still largely unknown. And if we're looking for the literature, we found the most relevant literature is talking about like TSC can produce some several molecules like growth factors, like messenger RNA, microRNA, or non-coding RNA to MSC through like extracellular vesicles. So they can provide this kind of several components to affect MSC function, including their proliferation and the osteogenesis. So they can improve the MSC function. But can TSC direct differentiate or trans-differentiate to mesenchymal lineage cells are still largely unknown. So that's why we start this project with Accelerated Bioscience company. So we get the cells from them and we try to compare the TSC with MSC. So we culture the MSC under MSC culture medium we regularly use. And also we culture the TSC as and using the TSC culture medium suggested by the company. And also we also try to use the MSC culture medium to culture TSC to see if we can using our MSC medium to promote TSC to become MSC-like cells under this culture. So we have these 3 groups cell culture, and we simply go to collect the RNA and doing the RNA sequencing analysis in using these 3 groups cells. So this is the data we got recently. As you can see, the left panel is the MSC we derive, we isolate from the orofacial tissues, mesenchymal tissues. And we have 2 groups TSC, like TSC1 is cultured in TSC median, and then TSC2 is culture, the TSC culture in the MSC median. And we can find when we compare the MSC with TSC1, it means TSC culture in the TSC median, we found around 4,000 genes are different their expression levels between these 2 types of cells. And if we culture the TSC in the MSC median, we can see large amount of genes, the expression level go back to MSC-like level. Okay, so you can see as I identified in these 2 areas, you can see the TSC1, they have highly expressed compared to MSCs in this area of genes. But after we put the TSC in MSC median, you can see if we're looking for the number, you can see around 1,000 genes change their expression level to MSC-like level. Okay, so this is overall gene profile between TSC and MSC and with different culture conditions. Okay. And if we use the pathway analysis to looking for these 2 types of cells, we found you can see for example, the growth factor signaling or like replication signaling are all activated in TSC cells. And also, very surprisingly, we found TSC also have highly expressed skeletal developmental pathway genes. So this is kind of surprising because we didn't expect the TSC have like a better differentiation ability without induction. But this analysis show like actually TSC even have expressed higher bone development or bone morphogenesis related pathway gene expression compared to MSC. No, and this is the you can see we compare MSC with TSC culture in the TSC median or TSC in the MSC median. They all have similar trends, like they all have increased growth ability and also have increased bone formation ability. So we try to focus on like a preparation specific pathway and the genes, and we found the cell preparation specific signaling, like Wen signaling, like TGA-beta signaling, and also like DNA replication, like a cell cycle. They all increase in the TSC compared to MSC. And this is another analysis we call GSEA. It's also kind of using some enrichment analysis in RNA sequencing. We show the cell cycle, like metabolic pathways, and like a MAP kinase pathway are also activated in TSC cells. And also we performed the in vitro study. We culture the cell to compare MSC and TSC, and using the proliferation marker Ki67 to stain these two types of cells. And you can see the Ki60 positive cell number are increased in the TSC group. And also, if we calculate the Ki67 positive cell divided by total cell percentage, we can see there are more cells in TSC group. They are under cell cycle or under cell proliferation status. Okay, so overall, this data show TSC have higher proliferation ability compared to MSC derived from orofacial region. And then we considering how the TSC in the differentiation status. Okay, so we culture the TSC and MSC, and in certain stage we change the median to induct they to become bone cell like osteoblast. Okay, so based on the RNA sequencing data, we see the TSC have highly expressed like a skeletal muscle development pathways and like a Wen signaling pathway, which is very important for bone formation, and also like a AKT-mTOR signaling pathway, which is also important to regulate bone formation. So you can see all these pathways are highly activated in TSC cells. And if we put the cell again in, as I mentioned, in the like osteogenic induction condition, you can see compared to MSC, the TSC have higher mineralized area after 4 weeks culture under this induction condition. And if we looking for the gene expression, you can see the bone-related markers like alkaline phosphatase and the key transcription factor runs 2. They are all highly activated in TSC group. So all this data suggests TSC have strong proliferation ability, and also they have strong bone differentiation ability. So next step, we try to culture these cells to compare with MSC and implant with the carriers, the hTSCP carriers, to the nude mice. And we are still waiting for this data. We want to see how their ex vivo, or we call like in vivo bone regeneration ability compare MSC with the TSC to see if they can direct bone cells in vivo in the nude mice. Okay. And in the future study, we want to translate our in vitro finding to the in vivo one. As I mentioned, we do a lot of cell therapy using disease model, for example, like autoimmune disease. We systemic infuse the stem cell like MSC into the disease model. They can through like regulate the immune system to cause immune tolerance, or they can transfer like cellular components through like exosomes to transfer cell component to the disease cells to rescue their disease from their phenotype. So we try to understand if TSC can have similar or even better like proliferation, differentiation ability compared to MSC. We will try to use our animal model, and we will do some like a systemic cell therapy using this cell to compare with MSC to see if they can have similar effect to cure the disease or for the tissue regeneration in the future or in vivo study. So I will stop here, and this is my lab members and also our collaborators from Penn, from NIDCR and from UCLA. So then my research are mainly supported by NIH. Thank you.